Well, hopefully nobody calls. This is the makeup recording. It's Saturday, the 14th. This is the one for yesterday, the 13th. I was unable to be there for any one of a number of reasons, but I'm here now. And so I actually did a little bit of this introduction before. And we're doing the peripheral nervous system. Our focus point, anatomically speaking, are going to be the eye and the ear as much as anything else from an anatomical basis. And I talked a little about these types of things and different receptors and proprioceptors and the different nerve endings. It really wasn't a big deal. So, but however, we do the eye physiology as part of the nervous system physiology later. Let me get over here. So, as you can see, it's extraordinarily, 70% of sensory receptors, half cerebral cortex. We only see a sixth of the eyes, and it's still very tiny. There's a lot of fat and things like that, and it's protected by the orbit. All those good things that are there, and the accessory structures as well. Well, we won't have to do uh, the muscles because you're already reviewing that for the exam. The LR6SO43. So you're looking at that for more than one type of a thing that's there. So you're going to be familiar with the extraocular eye muscles. But this is a good look at the eye itself. And even when we get to the anatomy test, I may or may not use this diagram, but just even though you're probably familiar with these things, I don't go crazy with a lot of this minutia, like the medial and lateral commissure. Okay, they're the corners. The bump that you feel on the medial aspect near your nose called the lacrimal caruncle. What's significant is obviously you have the lacrimal. Uh, that's more lacrimal foramen, or this tiny opening that's in that lacrimal bone that gives it away. That's there, and everybody knows what eyebrows are, and eyelids are, and eyelashes are, and all of those things. But really, to me, the big parts are what you may or may not be as familiar with, and that's this interior aspect. So the pupil, that small, roughly circular area that increases or decreases depending on the amount of light or if we're doing close work or looking far away. The pigmented area that surrounds it is referred to as the iris. Okay. And the pigment obviously varies genetically, and there's other factors. The white of the eye is called the sclera. And as you'll see, it's one of three what we call tunics. So just like as you'll learn in blood vessels where we talk about tunics, these are older terms. So the tunica fibrosa, the fibrous tunic, consists of the sclera, and it's mostly collagenous fibers and other proteins. And what happens is the arrangement is such that it's opaque to light, so we see the white portion. What happens here, once it reaches the iris and including overlying the pupil, there is a clear portion of it referred to as the cornea. So when you hear about corneal abrasions, it's not this part of the eye, but it's this part of the eye that you'll see there. And the tiny bits of red that you see there are really not, and again, this is not part of that outside tunic. This is the conjunctiva or conjunctiva, depending on how you pronounce it. It is normally clear and it's a thin membrane, but it has blood vessels. So whether you're struck in the eye or you rub your eye, you have allergies, and all of those things will tend to cause vessels to dilate or potentially to burst if you're struck in the eye, let's say by an object. Uh, and people rubbing their eyes, and obviously there's things like alcohol where it dilates the vessels, and we see bloodshot eyes, maybe after a long evening out indulging, those kinds of things that are there. But understand that that thin membrane is the conjunctiva or conjunctiva. So the lacrimal apparatus, just the tear apparatus. We looked at the extrinsic eye muscles. I don't have to go over it again, so I don't plan to. And yeah, the supraorbital margins that are there, that's where your eyebrows are. Everybody knows their eyebrows, what they do, who cares? Okay. The little folds we call the eyelids or palpebrae. So when you hear the palpebral fissure, it's the opening in your eyelids. The commissures are in the lateral caruncle that's there. And they have called them tarsal plates that anchor the muscles that are so common. As you get levator palpebrae superioris, lifts up your eyelids. Orbicularis oculi closes or opens your eyes, as you probably recall. And the eyelids, you can see blink a lot, and they're constantly, and we blink more when we get something in our eye. moistens the eye, and it sort of acts just like as a continuous lubricant. And most people, you don't sense it, but you are blinking that are there. You can, something catches you, or even a breeze in your eye. Obviously, you might blink with regard to that. They look at these modified sebaceous glands that they happen to call tarsal glands, and even then there's modified sweat glands as well. Again, not when I bust through things this fast, they're not major. It isn't a good illustration that I do occasionally use because you can see the clear cornea and the white, even though they've sort of lifted it up here just to give a demonstration, the sclera. Again, similarly made, but the fibers arranged differently. Lets one appear clear and one have light. So that in itself is kind of interesting. And when one of those glands, I mean, sometimes it's, I don't even know, I'm sure the chalazion, but what's interesting, so that usually is not a big deal. Where styes, and if you ever had one, and I don't know how common they are today. Oh, they are annoying. These are then with the eyelashes, and it's just like it's almost like a pimple or a pustule developing in that kind of. It's really unpleasant, and sometimes you have those lands back in the old days. I don't know if they're as common today now as they were. I think we're so much more conscious of hand care and not rubbing the eyes. And the little kids would get them because they rub their eyes so much. So the conjunctiva or conjunctiva is transparent. Okay, it has some lubricating ability because of the lining that's there. It's interesting. It's got a layer under the eyelids. It has a layer that covers the whites of the eye. So it's almost like when you think of a joint lining, it's kind of the same way. It covers the bone, and then it kind of almost doubles back on itself, so to speak, in the area around the joint itself. Okay, and it's easy enough to see. And there's a little sac, and that's about all you have to know. And certainly, when the redness increases against a white background, depending on the severity of it, we can go from pink to really significantly red, or black and blue if somebody struck in the eye or has a broken nose or a sports injury, those kinds of things. But pink eye is annoying. Again, I do not know how much in depth you went it with Dr. Maurer in micro, but pink eye is about a third of the time they're viral, and this is true for ear infections and sinus infections, and about 2/3 of the time they're bacterial. They're very contagious, and the trouble is, and it's true with all viral inflammation things around linings or membrane, just like viral pneumonia. Viral infections weaken your defenses, and so you're more predisposed to get bacterial infections, and they're much more destructive because the viruses will go away, but the bacteria, because of their actions, it's less of an immunological response. There's more destruction because they typically release toxins, which is sort of how bacteria do the bad things they do in many instances, not entirely, of course. And because of that, you're always worried, even with some of this viral conjunctivitis or viral ear infection or sinusitis. We use antibiotics anyway to prevent the bacterial infection. It's like I was telling you, maybe I did, I'm sure I did. My son getting uh, particles in the eye inadvertently from his likely future stepson, uh, who squeezed something. And I'm like, "Well, what do they do?" But he, A, misses 2 days of work. B, that eye immediately, the next day, started to really have a lot of productive stuff. That's when he had to go, and he got the eye drops in it. And because it heals, you're generally looking at a 24 to 48 hour turnaround. And yeah, he'll be fine for Monday. He was probably pretty good on Friday, but took the day off anyway, just in case. And so here you have the lacrimal apparatus. And what's interesting about this, the gland is sort of superior and lateral, and the collection of the opening in the medial commissure is inferior and medial. So it kind of, you can see it's got a lot of good stuff in it. It's like a dilute saline solution. It's got antibodies. It's got lysozyme in it. So it's got a lot of stuff in it. And so it drains into that duct in that tiny opening in the lacrimal bone. And you can see it. It's a great illustration because what you're looking at here, it is, and the fluid is released there, and by blinking your eye, it sweeps it down, and eventually it's collected here and typically drain into your nose. That, again, that's one of the interesting things why nasal infections can go to your eye, because the fact that tears usually go this way doesn't preclude bacteria or other problems coming up that way. It's sort of the old two-way street kind of a situation that's there. And the analogy I always use, whether it's clever or not, is like it's like the wiper you have in the back if you have like an SUV or a station wagon, or like I have a van. It's like it's so a lot of times they spritz from above and then the wipers spread it around. It's kind of like that. Spritz from above, wipers, and eventually drains out there. So that's, and again, here you can see about the inflammation and lacrimal mucosa and watery eyes. And so a lot of times they're intertwined. So when people have these things, we see a lot. That's why kids get a lot of that stuff. They just do. Oh, okie dokie. So I just figured you'd know about that. These are the extrinsic eye muscles. You can buzz through these, the LR6, SO43. It's going to certainly be relevant. These are very similar. They show what they do. And you can see that one. It was hard for me to draw it. But superior oblique, what it does, because it's on top, it's going to rotate the eye at an angle downward. So it's going to come, the eye's going to go down, but because of the angle, this is going to make basically the pupil and the visual parts sort of rolling down externally like that. It's interesting what they can do. And so you have to follow the muscular activity. It's a good look. And here are all those things. And this is the whole idea of double vision. When they aren't perfectly coordinated, it really affects us with our ability to focus. And there's lots of things that can happen. Weakness in the muscles, neurological disorders, certainly injuries. Uh, and we do it more when we see it with concussion. It's a little bit more about the processing and the message being sent. And certainly it could point to damage in the orbit itself. That's there. Cross eye, a little bit different. Okay. Not quite the same. Probably has more to do with something that has, again, you can be born with this. It can develop again from injuries, nervous system disorders. And again, there's a lot of variation. Maybe I told you, or if I did not, that same son with the eye, he had a mild degree of cross eye. We were concerned about it. Took him to the eye doctor, the ophthalmologist down for Children's Hospital, right down in Oakland, and he looked at it. He goes, I was like, we don't have to do anything. We'll try some exercises, and he's probably going to end up having to wear glasses. I said, what about, in general activity? Because he's a very active kid, loves sports. He goes, a very famous tennis player who won 3 French Opens and 3 U.S. Opens, Yvonne Lendl, during the McEnroe era. He goes, and he said, you know who he is? And I said, of course, I'm a big sports guy. He goes, he's got one that's much worse than your son's. We don't know. Nobody operated on him. Okay, case closed. This is the anatomical part. I'll show you in the lab, not this week, but obviously next, when we'll sort of consolidate a lot of this stuff together as we start the heart. I'll be able to use the eye and ear models, and we have a number of them. There are 3 layers. So now we just call them fibrous, vascular, and the inner, which is the nervous layer. In the old days, it was tunica fibrosa, tunica vascularis, and tunica nervosa. And again, we use the old terminology for fluids. Fluids were called humors. So when someone was again, this is good. In the old days, we looked at phlegm and blood and bile and black bile. And so if somebody had a biliary, they were sort of a nasty person. They had a dry, not a lot of emotion. They were very sanguine. They were just they were sort of phlegmatic, that kind of stuff. So it's interesting if you go back and look at those, and that was the origin of bleeding people, feeling there was an overabundance of blood in certain areas. And so you have, as you will see, when we do the eye dissection, which is our next big dissection to do, it is just oodles of fun. I always call it the best 15-minute dissection there is, because you see everything. And the lens divides them almost to the same way as the visual portion of the eye that you and I can see on somebody else versus the part that's within the orbit. It's about 1/6 to 5/6. It divides them into 2 chambers, the anterior and posterior chamber or segment. And again, what's very, very interesting about that is, pardon me, is that the fluids are different. So there are two isolated chambers. The anterior section has an aqueous or water based fluid. The posterior section is a gel like material fluid, but they call them humor. So you have aqueous humor and vitreous humor, and sometimes because of that gel sort of, it's almost like a little packet. They will sometimes call it the vitreous body, and by definition, they're clear. And the vitreous is interesting. This gel-like area of interest forensically. Sometimes it'll reflect because we have it from the time we're born, and it will. If you can sample the fluid post mortem for some clinical clues about cause of death or maybe disease entities that might have got accumulated within that. That's one interesting thing. But the aqueous is constantly made and constantly replaced. You make a little bit every day and you drain a little bit every day. That's the good news. The bad news is if you make too much or it doesn't drain rapidly enough, it puts pressure against the lens and creates a very painful condition that can lead to blindness. It's very famous, called glaucoma. Most of the time, it's a drainage related problem. And thankfully, there's very good treatment that's available for it. So this, and we have a model that does the same thing. It's a great illustration. I probably use it on practicals more than once because there's a ton of things. So you can see the continuous outer fibrous layer has the sclera, which is white, and it becomes clear as it transitions just at the start of the iris, this pigmented portion, and ends on the other side of the iris. And you can see the pupil, obviously, that's there. That's the constituents of the fibrous layer, sclera, cornea. The pigmented layer is this darker layer that you can see. And interestingly enough, probably its major role is to prevent what's called scatter. So it's like anything else. In the old days, before we had digital photography, we used dark rooms. And to develop things when I was started in practice, pushing 44 years ago, uh, when we took X-rays in the office, I had a little closet with a red light that was light and tanks there, and we developed the X-rays by hand. We dipped them, and it had to be darker; you'd lose it. So that and the whole idea of using a dark room or taking out film when you took it to be developed at a photography store or a grocery store, wherever you took it, was a lot of times that they had to make sure it wasn't exposed to light because you'd lose it. That's the scatter issue. So scatter is a confusion. Okay, it's like when somebody turns the lights on after they've been off for a while, and that opening a light before your pupils can adjust. Visual confusion, a lot of time unpleasantly so. So that pigmented layer plays a role in that. So we have this vascular layer, and what you're going to see when we get into the retina, and we'll look more closely at that in physiology. There's actually an internal pigmented layer of the retina, though you really can't see it from this illustration. That effectively is where the light actually goes through this membrane and doesn't scatter because because this protects the vascular layer, and then effectively this is what triggers our receptors called rods and cones, and we are able to see because of it. The continuation of this vascular layer to continue with it is it forms a ganglion that's here. This is the parasympathetic feature, the glandular feature. And from this, we call it the ciliary body or ciliary zone or ciliary ganglion. What extends from it are, as you can see, the iris, and the pupil is within it. And indeed, these suspensory ligaments, little strands that act like ligaments pulling on the lens when when the pupils dilate, when they widen, okay, because it's somewhat of a circular action. Again, the way it's almost like one of those orbicularis style muscles. They open and close. When you open and close this opening, it will tend to cause the lens to flatten out. And when the pupils constrict, then the lens becomes more rounded. So it changes the shape of the lens, and that changes the way the light enters. When you're doing close work, the light, because of the roundedness, is able to very, very easily direct this, or sometimes if you wear glasses or contacts, whatever, to the area where you see the best, and that's called the fovea centralis. That's located within the macula lutea. That's the highest concentration of your best visual cells, which are your cones. Okay. So these structure, and when you're doing distant vision, it flattens it out. It doesn't have to bend the light to get there. It almost naturally goes there. But when you're doing close work, that roundedness of the lens changes that angle. That's physics, and we know I don't do physics. That type of thing. It's interesting. The edge of this sort of ciliary zone creates a almost like a serrated edge. They call it the aura serrata. And what's neat about that, particularly when we get around to doing the dissection in the eye, you see it in either the sheep's eye or the cow eye. You can see that edge. You can see on the lens the little remnants of the suspensory ligaments. You can see all of these structures that are there. That's why I think it's so fascinating. And then within this, and then we get to the nervous layer, nervous tunic. And the interesting about it is that, remember, these are sensory nerves. You have sensors everywhere except here. All the little sensors. And sometimes you get lucky in the membrane that you open on the interior of the eye. When you actually look at the retina, you can actually see the tiny fibers collecting here, what we call the blind spot or the optic disc. There's no sensors, there's no rods, no cones, but all the nerves carrying those impulses sort of meet here and exit. And so then in this case, the nerve itself surrounds it, the nerve, and the interior, interesting, is the blood vessels themselves, the artery and the veins are within that, and they're trying to depict here the vitreous body. So sclera and cornea again protects, shape the eyeball, place for the eye muscles. That's all about the sclera. The sclera is continuous with the dura. The cornea, transparent, clear window, covering, and there's epithelium on it, corneal epithelium, and sodium pumps maintain that being nice and clear. So they're able to maintain that because if they stop working, the eye will cloud over, and that's exactly what happens when somebody dies. And so when you die, those pumps, there's no more ATP you're making to make those. And so it's very disconcerting um, to see somebody in repose, and, relative, and their eyes cloud over. So typically they make sure the eyes are closed. And they get a lot of pain receptors. The vascular layer, another name is the UV. I don't use it. Another name I do use is the choroid. That's the bulk of it. The ciliary body, and the continuation is the iris. The choroid, the substantial posterior and portion about the 5-6, blood to all the layers of the eyeball, brownish pigment which scatter. Okay, the ciliary body anteriorly rings around the lens. So collectively, we call it ciliary muscles. They exert, you're not muscles in the classic sense of muscles, but they exert a force on the shape of the lens by way of these suspensory ligaments that are there. And then the iris, the pigmented portion or colored portion of the eye. Pupils just the opening, and again what it does. And there's also, you can see here, pupils dilate when it's emotional, dim light, they widen, as well, bright light, they constrict, etc. We went over that. And this is showing the rotational thing when they constrict versus when they dilate. What happens? That really, it's almost like, imagine that stretching out the ciliary so it flattens out the lens. When they constrict, it closes those sort of ciliary muscles. So it makes them less taut, and so the lens can go back to a more natural state of being more rounded, if you will. And what you're going to see, the clarity of the lens is affected because obviously, whether it's a cow eye or a sheep eye, they're deceased. And the lenses are very clouded over, not clear. When you get clouded lenses or clouded cataract, clouded cornea, and I just said the word, effectively those are areas where we see cataracts. And cataracts are just that, opacities, areas that where the clarity or the clear nature of those structures has been, for whatever reason, lost. Okay. And again, that's a really interesting thing that happens in that regard. So you will see that. And again, if I didn't tell you, I'll tell you again. Cataracts, there's a lot of different reasons. I'll go over them here. But by and large, it's an aging phenomena that a lot of people get when they get there. There's certain things like diabetes and smoking, and certainly there's hereditary factors that are involved. But by and large, if you live long enough, it's something that you struggle with. So the retina, which is really, if you remember when we looked at the brain earlier in lecture, those little outpouchings coming from the brain were really the retina developing. So it's really, again, it's really the extension of what is the second cranial nerve. Photoreceptors, and it's all about transduction, changing it from photo signal to an electrical signal, different kinds of cells. And again, it has this outer, close to that vascular layer, pigmented layer, and the inside is the neural layer. And the neural layer is actually where the visual impulses are processed. And there we are coming back to the optic disc. So the single cell next to the choroid, again, more external, okay, that's where it again plays a role in scattering, but that's where the light signals, the frequency of the light is absorbed. Okay. And it also plays a role, as you'll see when we get into the physiology, we're constantly we're using and breaking down and remaking these pigments in the rods and the cones. So it helps to gobble them up in that layer, and vitamin A is the big player in this. So the neural layer, more transparent, anteriorly to the margin of the ciliary body, okay? Interior end has serrated edges called the ora serrata, as you could see. So you have three different kinds of cells that you'll need to remember. And basically, the photoreceptors, the rods and the cones, they're the ones that capture the light and wherever they're exposed and location, what you're seeing. That triggers the first, that causes, what you're going to learn is that the light causes a shape change in that pigment. Okay. That will then effectively fire bipolar cells. We only see bipolar cells in special senses. It's really like what makes special senses special, so to speak, is you get two kinds of sensory nerves in sequence. You have one receiving the photo signal in this case, and then another one taking that signal, which was light oriented as a photoreceptor, and basically converting it to an electrical signal. And then the ganglion cells collect this and they eventually feed in to wend their way down by way of the optic disc or the blind spot and effectively form the nerve, the optic nerve. So that's where it all lives, so-called blind spot. As you can see, 250 million photoreceptors, and they're one of two types. It's about 20 to 1 rods to cones. Rods predominate. Rods are for grayscale, non-color vision, so to speak. They are much more sensitive to by design. So in less light, we can see very well with them. At night, when you go outside, that's what helps you see. They're highly concentrated, and anatomically, they're much more on the exterior or the periphery rather than centrally placed. Color requires a lot of light, and they're very, very sensitive receptors, but they need to have light. So the rods work all the time, even in low light, and some people have remarkably good night vision, not yours truly. Okay. So dim light and peripheral vision, that's why, because they're focused around the periphery of the retina. That's why you and I, well, you'll catch a glimpse of something at the side of your eye. You're more likely to see a deer coming from the side of the car than you are one standing in the middle of the road, or any other animal for that matter, because you just don't see them, particularly at night. Okay. No color vision. The images are not sharp. They're somewhat fuzzy. That are there. The cones, very high resolution. It's just like having, where you have to have the right lighting to get a really good picture, whether you're using a cell phone camera or your own camera. Okay. The macula is the area posterior pole, slightly lateral to the blind spot. It's almost all cones. And the tiny pit called the fovea centralis is all cones. Okay. And so and our eye movements allow us to focus there. Corrective lenses focus it there. This is the illustration you'll see. You can see the rods because they end up looking rod shaped, and the cone because it's got this sort of a pointy area at the end. And again, the rods are much more numerous. These are the bipolar, these are the ganglion cells. And the ganglion cells, that's going to be the directionality of the optic nerve. And it's what it looks like microscopically. So there's the photoreceptors from the choroid area, that central artery and vein that I pointed out before. That's the inner 2/3. Okay. And this is the neatest thing. These are all tiny arteries. So they're basically they're arterioles. And arterioles are extraordinarily small. Okay. You can't see them with the naked eye except against the retina. And this is what we look for. So nowadays, in the old days, we always used an ophthalmoscope. It took some talent and some practice. You had to dilate the eyes with some type of chemical agent, an eye drop that would cause the pupils to dilate, so you could get in there and take a look at what we call the eye grounds. Really, because an arterial in the eye, and arterial in the kidney, and arterial in deep within the tissue or in the intestines all look the same. They look like this. So if one is damaged here, you can make the assumption fairly easily. It's not a big leap to say that the ones in the kidney, and that's what we look at them for. Because if we see narrowing, if we see areas where, and the heart for that matter as well, I should have mentioned first, if we see where it's not where tiny little nicks or irregularities or points or bends that are kind of sharp, all of those, that's bad. You'd see it. That's what you look for in diabetics. That's what you look for in smokers. That's what you look for with a lot of disease entities. So it's really important. What the difference now is that the technology has improved so much that most time when you have your eyes examined, they all have a machine that's able to give you, they'll load you on a flash drive the picture so you can see it yourself. But you can get a lot out of it. And the anecdote I'll share with you is when I was a heavy smoker, I remember having an insurance physical with a family doctor. And he looked at me, goes, I know you're a smoker. He says, but I'm starting to see some funny stuff in the eyes. He says, I wanted to make you aware of it. It's not anything that's going to affect the insurance abilities in the early stages, but you do need to be aware of it. Okay. Well, years later, within the last 10 years, and I'd stopped smoking, like I said, about 15 years ago, I had an eye exam over here very close to where I live for a new pair of glasses. And son of a gun, the doctor looks at it with this newer technology, and she looks at me and she goes, you were a smoker. She says, because I think you had problems here, but now they're getting better. Go figure. Pretty good for technology. And so this is a retinal detachment, and the vitreous seeps can lead to blindness, traumatic blows, sudden stopping movements, obviously bungee jumping, but something more, any kind of thing like that could potentially affect it significantly. So there's a lot to this. And you can see what they're talking about. And nowadays, there's a greater possibility to repair them. Eye surgery has improved light years in recent memory. It's just traumatic. I tell all students, I had a history teacher who was one of our, we were on the debate team, and there were two history teachers, and he was one of the two guys who were on the debate team. They kind of ran it. And really nice guy, wore those big, thick, heavy glasses. So this is back in 65, 67. So it's a good, we're pushing 60 years ago. And he had to be sandbagged. He had a detached retina, had to be sandbagged in bed on his back, unable to move for 10 days. And they did the surgical correction because he was losing his sight. A young guy, probably in his 40s. So the segments, again, the posterior segment separated by the lens is the vitreous area. That's what keeps the roundness of the eye. Where obviously the shape is secured by the sclera, but this kind of fills in that area and makes sure that the retina is pressed appropriately against the vascular layer and held in place by the fibrous layer. And the anterior segment is the one with the aqueous. So vitreous, light goes through it, holds the retina firmly, supports it. Intraocular pressure is part of it, forms in the embryo. It's for your whole lifetime. The anterior segment, okay, and so that's it has two segments, but think between the lens and the cornea itself. Aqueous humor, okay, continuously formed, little capillaries. There's a canal that's within that area of the eye. They have an illustration, so I can't really point out where it is, called the canal. Famously, the last name is Schlem. Okay, and all of these things, yeah. And so there's a lot of stuff, nutrients and oxygen, mainly to the lens and the cornea, but also to the retina, and removes waste. And they show it to you here, and you're going to see this little canal that's in there. And you can go through all these if you want. just fine. And that's the idea of glaucoma, in dangerous levels compressing the nerve. There are eye drops have been used. I know many people who have had this, and again, that's the intro. And again, they do the same machinery. Once you reach a certain age, unless there's significant hereditary factors that are there, and again, there's surgical intervention if necessary. The Again, the lens is biconvex, so it's rounded, so it's convex on either side of it, so to speak. And again, you have epithelial cells and fibers. A fancy protein is called crystalline. And again, it changes and it's less elastic, less stretchy with age. So most of us, we get older. There's a lot of significant visual problems. That's what's called presbyopia. And this is the clouding of the lens. Again, these are the cataract formations. We used to think vitamin C was very, very safe, but now we can see people used to take excessive amounts because they thought it was the cure for the common cold, would prevent common colds. It's because a two-time Nobel Prize winning laureate, Linus Pauling, said he hadn't had a cold in 5 years and he took mega doses of vitamin C. That would happen in the 70s. Everybody started taking vitamin C like going crazy. But now we have lens, we can implant lens, we can remove cataracts. They're done very, very rapidly. I told you about that being in bed for 10 days. Nothing like that. Thank you very much. Right now, I mean, they'll send you home the same day with eye drops and just tell you not to lift anything. That's about the size of it. So I will stop it here. Okay. I will show you just because we finished this particular one. And I did want to show you what B starts with this is where the optics come in. So I just want to show it to you before I conclude. This is all that stuff. And so you're all the closing and the accommodation and the different sidedness. And this is when we get into it in physiology. We'll get into actually how these all work. That's what this particular PowerPoint is devoted to, nearly exclusively. So it's not like we're skipping it intentionally, but to me, it's much more physiological and the different pathways to the brain, et cetera, et cetera. I think it's all that. And it isn't until the very end that we'll get to smell and taste. And to me, I think that they're more physiological than anatomical, at least for our purposes now. And the last part, and the same thing with taste. The last part that we'll do is going to be C, which I won't click up here. And because it will show the ear anatomy, and that's the other important part to go through. So thanks a lot. Again, I appreciate your indulgence since I wasn't able to be there on that particular day. And hopefully you get a chance to listen to this, maybe probably before Wednesday, where we probably will get back into the ear. Actually, we'll probably do a little bit on Monday. Bye-bye. Thank you.